Yeah, it's a great pleasure to be here. And I have a sore throat, so bear with me. Sometimes I might have to go like this. Yeah, so I went to the SETAC meeting in Hobart, and Kat suggested I come up to Melbourne, and that's great. I'm on sabbatical at the University of Newcastle. A very different subject, but it's always related to reproduction for me. So I'm working on amphibian reproductive biology with a herpetology group there in the University of Newcastle. So today, there's different titles to this kind of talk, and I think pollutants are affecting more than our hormones. And the whole concept of endocrine disruption is now expanded. And the point today is going to be to think about the brain as a target. Now, the brain is the major source of hormones. Control all of the bodily processes that you kind of care about, reproduction, growth, metabolism, things like that. So that's the kind of message for today. So we'll just get on with this. Is this recording? Not yet. Okay, better put it on. They want to record this stuff. Is it working? Yeah, okay. So it's a small group, so we can be kind of casual. So if you guys want to have more of a discussion, if there's something that you want to talk about right in the middle, that's cool with me too. So let's see how this works. So we put out a couple of definitions of this emerging concept called neuroendocrine disruption. Now, it's the same idea as endocrine disruption, but the last 20 years have really been focusing on how things like sex steroids, estrogens from the pill, for example, would affect the liver, or would affect the testes or the ovaries, and much to the kind of forgetting about the brain. But it's hormones in the brain that control reproduction. So the idea in these review papers here, the first one in 2011, was to kind of say, hey, the brain can respond in milliseconds to a stimulus. When you blink your eyes, light. When you see your ex in the hallway and you get embarrassed, that's milliseconds of hatred, and then it turns to aggression. But it's quick. The brain acts quickly, and then the hormones catch up later, and the ovaries and the testes and all that catch up later. So the idea that we put out some years ago is to start thinking more about the brain as a target for these pollutants, and that's really kind of the message. And I'm going to give you 3 examples. 2 from my own work, number 1 and number 2, and 3 from colleagues in the U.S. So I'm going to give an example of a common neuroactive pharmaceutical. It's called Prozac. It's one of the antidepressants, and it's a major pollutant, and it upsets hormone systems. I'm going to tell you about pulp and paper mill effluents. They actually have phytochemicals in them, and those phytochemicals can mimic brain neurotransmitters. The third is bisphenol A. A lot of people have heard about this one. And this is quite profound in my mind, how bisphenol A can actually upset many systems, including sexual behavior, mate selection, even now linked to obesity. So I've asked some colleagues to let me have their data. It's all published, but it's really their work. So antidepressants, you may know about these. A lot of people take antidepressants. In Canada and the U.S., it's around 1 in 10. There's big ones like Prozac. This was the first generation of antidepressants. The chemical name is fluoxetine, and its mechanism of action is called SSRI, selective serotonin reuptake inhibitor. So depressed patients don't have enough serotonin in certain synapses. So if you take this chemical, it blocks a certain transport protein, and it keeps serotonin around a little bit, and after some weeks, you start to feel a little bit better. It's been largely replaced by a bunch of others. There's 5 or 6 big ones. Some of them are shown here. What's amazing about the internet is you can find anything. Like some crazy guy made this slide. I didn't make it. So I just put in, Prozac and something, and I got this nice slide. But there's a lot of people on it. That's the point. And you can find it in sewage effluents. And the fun thing about Prozac from an ecotox point of view is that it's resistant to degradation, and it hangs around a little bit. And of course, we take it every day. We flush the toilet every day, so it's being added to the environment, even though it does degrade. It's pseudo-persistence. You're always flushing, so it's always there and up and down. So that's the idea here. So it's a reuptake inhibitor of this brain chemical serotonin. It's really used mostly for depression, but also some forms of anxiety, bulimia, and obsessive compulsive disorders. It's resistant to degradation. It can hang on to the sludge. And it's highly variable concentrations of the single antidepressant, but a high level in wastewater would be up to 0.5 microgram. So that's quite a bit. If you add up the top 5 of antidepressants, let's say in a small town of 50,000 in Canada, close to Toronto, this would add up to 1 microgram per liter of all SSRIs. So there's a significant amount of this type of chemical in our sewage effluents. And in any country using antidepressants, I suspect it's going to be anywhere between 0 and a couple of micrograms. Quite a few years ago, a visiting student from Germany came over, Jan Menigan. He ended up coming back for PhD. And we were just starting to get into that idea of Prozac as an endocrine disruptor. And we developed something called a microarray. Does everybody know what a microarray is? It's a way to measure thousands of genes. We use goldfish for a lot of our studies. And you can measure, in this particular platform, 10,000 genes at a time. And Jan gave injections, sort of mimicking the clinic, not the environment. And many different things changed. They changed a little bit, they changed a lot. And there was no super pattern. There was a lot of processes affected. And that makes sense because serotonin is in many, many brain circuits. But one jumped out at us, and this is called isotocin. And isotocin is a 9 amino acid peptide, and it's equivalent to oxytocin in our own bodies. Now, you may know oxytocin as the one that stimulates milk release when the baby cries. Oxytocin is involved in birth. Oxytocin is the defiliative hormone. You know, when you're feeling nice about the opposite sex, your oxytocin neurons are doing that. If the mom is attracted to the baby, it's because of oxytocin. It's a reproductive behavior hormone. In fish, it stimulates some aspects of spawning behavior. So this just jumped out at us. Severely decreased levels in the one part of the brain called the hypothalamus. And at the same time, we're a part of a consortium, Canadian and European scientists working on SSRIs, and one colleague from Guelph, Glenn van der Kraak, he was giving higher levels to zebrafish, and he was inhibiting ovulation. They weren't laying eggs. So we said, I've got the spawning peptide, and he's got no egg laying. We've got a story here. So we started thinking more about this. And here's just some real data. This is a goldfish brain. So this is the front of the animal, like this, and this is the olfactory system and the forebrain and the hypothalamus. And these neurons that control sexual behavior and contain isotocin are located kind of here, and then there's a minor population here. So this PCR data shows you what happens when you give 5 injections of Prozac over 2 weeks, every couple of days. Is severe decrease in the messenger RNA for this major important peptide. So this is the kind of things that we were doing in those days, and really led us to think about how Prozac could be upsetting sexual function. Now, here's an incomplete slide showing quite a few different studies, but it gives you the idea of how this is fitting into the environment. We know something about the toxicity of Prozac. It's basically not toxic. This is huge amounts. You would never, ever find this in the environment, never. We wouldn't take it if it was toxic. So it's basically not very toxic from the traditional kill 'em, count 'em point of view. And all the way down to nanograms per liter, you can disrupt sexual behavior in fish. So the studies I'm going to tell you about are in this range here, in the mid nanograms per liter. Okay, so we use around 500 usually, which is kind of a high environmental level. So Prozac and sex, could you believe it? in the privacy of your own home, please do this. You can Google those 3 words, and I did it in September, and I got 2.4 million hits. The number of hits varies if they clean up the double hits and all of that, but it turns out that in humans, one of the major side effects that people report is decreased libido, and this is for men and women. So there's something there. Prozac is not necessarily good for our sexuality. A lot of people talk about, you know, how they don't feel active and things like that relative to the other sex when they're taking Prozac. So in the original little pamphlet that came with Prozac and the clinical data that they report with that prescription drug, they talked about 4% of people having issues with libido. It's much, much higher than that. We know that now. So there's something with Prozac and reproduction. So in terms of an experiment we've done, and it's a complex experiment, so I'm going to take you through some basics, and then I'll give you some data. So in the goldfish, the female communicates with the male at the time of breeding, in all fish, in fact. So here's her ovaries developing. The oocytes are getting ready for ovulation. And there's a hormone from the pituitary gland called LH, luteinizing hormone. It's the same one in humans that would drive ovulation or sperm testosterone production, things like that. It goes up. It drives the last parts of oocyte maturation and ovulation. So how does the male know that she's going to ovulate? It's through her sex pheromones. So she is producing different steroids that have effects in the body, and they're actually released under control into the water, and they become pre-ovulatory pheromones. She signals specifically through chemicals to tell the male, he detects it with his olfactory system, and it says, hey, get ready, because in a few hours, I'm going to ovulate. So you better be ready. And he gets kind of friendly with her, he starts chasing her, and his LH levels go up, his milk, so the sperm and the fluid starts getting ready to be released. So this is what you can squeeze out of the male. Then she ovulates in the middle of the night sometime, and then all these steroids stop, and she produces another kind of pheromone called the post-ovulatory pheromone. This tells the male, you better get ready, I'm releasing my eggs. I'm going to ovipositive. So you better release your sperm at the right time. So it's all about spawning synchrony. And this is a prostaglandin F2 alpha. So within the female fish's body, this controls her oviposition behavior, and she releases it, and it controls his courtship and spawning behavior and the ultimate release of sperm at the right time for fertilization. So what I'm going to do in this experiment is I'm going to replace the female with just the pheromones. I've pre-exposed the male to Prozac, 2 levels, and I'm going to give them a shot of the pre-ovulatory pheromone and a shot of the post-ovulatory pheromone, and I'm going to measure milk. So the question is, does pre-exposure to Prozac affect milk release, the major male behavior, the one that counts? And the answer is yes. So fluoxetine actually inhibits sperm release in response to sex pheromones. So here's milk volume, and here's a bunch of different treatments, and I'll take you through them. So we exposed them for 2 weeks to fluoxetine, and then to control all these different drug combinations, and then you squeeze out the milk, and you measure the milk volume. So in the control, that was about 15 microliters, and if I give the pre-ovulatory pheromone or the post-ovulatory pheromone, I get a 2- or 3-fold increase. This is classic. This would be 6 hours of exposure to the sex pheromone, which would kind of mimic what happens in the pond or the stream, where the female is releasing her pheromone, the male starts following, and then eventually they're going to get it together and fertilize eggs. So if you expose them to a environmental or a pharmacological level, you kind of get the same effect. Basal milk production is decreasing, and the response to the pre or the post-ovulatory pheromone is essentially knocked out. So if you just focus here for the sake of time, you get a 3-fold increase, and this is basically bottomed out. The males don't release their milk. So we looked at the testes. The testes look normal. It's only a 2-week exposure. You wouldn't expect too many morphological changes, but they have lots of sperm. The testes look good. We did some gene marker type stuff. There was a few things changed, but really, they have sperm. They just don't release it. So for me, this is quite profound. All they have to do is be exposed for a couple of weeks, and they won't be able to breed very well. So that was one kind of result. There's some hormonal data that goes with this as well. It's not on the slide, but basically in the male fish, estradiol levels go up and testosterone levels go down. So we could see the molecular correlates of this in the brain as well. The estrogen receptors are changing. The male fish actually produces more egg yolk protein, vitellogenin. Of course, egg yolk protein is supposed to be for females. And that's as a result of this change in estrogen. So their whole endocrine system is upset just by exposure to a single chemical, fluoxetine. Any questions? It's all good? I forgot to ask about your background. Everybody's kind of from different disciplines, different backgrounds, mostly toxicology or any physiologists, mostly ecology, toxicology type. Okay. Another thing we've done is to look at food intake and growth. So here's an interesting study. Fluoxetine does more than upset reproductive processes. It actually disrupts food intake and weight gain. So here is animals in a period of the year where they are not reproducing, but they're growing, starting to grow, post-reproduction. And over a couple of weeks, a control animal would gain about 1 gram more. They weighed about 15 or 20 grams, I forget exactly, at the beginning of the study. And our positive control here was to deprive the animal of food. So over that period, they lost about 3 grams. If we give, should be 540. If we give the environmental level, they don't gain weight. If we give them the high pharmacological dose, they lose weight. It induces a fasting-like response. And it's really hard. We haven't figured out if it's a decrease in food intake specifically that's causing this, or is it fluoxetine doing something to the metabolism? The answer is that, in fact, it does both. They eat less, and their metabolic processes are also upset. And I'll show you a little bit about that. So in fact, they lose weight. I do believe some people take Prozac to lose weight. I don't know if I think it's probably off prescription. I'm not sure exactly. It wouldn't be something that you would want to recommend to anybody, given the other side effects. No food, no sex, no fun. So not something to really do. So we've done a few studies on this, and the response in terms of metabolism is quite global, if you will. The reduction in food, feeding, and weight I told you about. Circulating glucose levels decrease in these animals. Gluconeogenesis in the liver is decreased in these animals. And the feeding circuits, the neuropeptides in the brain that control feeding, are also disrupted. The ones that tend to stimulate feeding go down. The ones that tend to inhibit feeding in the brain go up. So this is a pretty upset animal in one sense. Now we turn to the zebrafish for a study of the liver to try to understand how something that acts in the brain could actually upset metabolism. So this is the downstream effect of being exposed to the brain and Prozac and what have you. And a postdoc in my buddy's lab, Paul Craig, said, hey, let's use the zebrafish for this. Because, the genome's annotated and we have lots of tools and things like that. So he did a really interesting study in my mind, and he found that all these types of signaling pathways, things related to metabolism, insulin signaling, adipogenesis, glycogenesis, triglyceride synthesis, these things were all upset. And the way he did this was to study something called microRNAs. Have you guys heard of microRNAs? A little bit. So these are really cool things. I'm going to give you a slide in a minute, but they're short RNAs that actually regulate other RNAs. So it actually is a negative regulator of messenger RNA, of the thing that would make the protein. And what really jumped out at Paul and the rest of the team was that some of these microRNAs that are known to inhibit AMP-activated protein kinase, that's a fancy name, for a super important metabolic enzyme. So they were actually increasing this, that would inhibit metabolism. This is the global metabolism regulator. If you look it up on Wiki or on a reputable site, it would say about the same thing. This is the master metabolic regulator. And what are microRNAs? And this takes a little bit of explanation. So here's your normal messenger RNA that would code for a protein. Let's say that subunit of that enzyme. And a microRNA is kind of a mirror of one part of this. And it basically will bind to the 3' untranslated part of the mRNA, and it's essentially going to induce degradation, okay? Or through a couple of mechanisms, it actually can degrade, so this just disappears, or this can actually repress translation. So the message stays there, but you don't get the protein. The most important thing is that if you have an increase in a specific microRNA, you have a decrease in the target. So Paul had screened hundreds of these, and he found a few that were really important for metabolism. So here we're having something acting on the brain, doing all kinds of things, and also upsetting liver function. Okay, so this is the master regulator of energy homeostasis. And that's a quote from, kind of the gene ontology websites when you go to try to find out what a gene is doing. So this is an important molecule. Okay, and this is in every cell. It's especially important in the liver. So two of these, it doesn't matter their name, they were both very highly upregulated, and they're involved in gluconeogenesis, regulating cholesterol and fatty acids, what can be more important to metabolism. So Paul had developed this really fancy method to detect 200 of these at once, and what we found is that fluoxetine is essentially making a fasting-like response. So just fluoxetine alone makes you kind of fast, and when we put the two together, fasting alone and fast plus fluoxetine, we have 200-fold increase in these very important small microRNAs. They're like 19 to 21 nucleotides long. And he did a correlative study for these two things that are attacking different parts of the pathway. And here's the fasted, here's the fed with fluoxetine, and here's the fasted with fluoxetine. So it's a serious decrease in the expression of these things, of these particular subunits. So basically, we're inducing a fasting-like response to these animals. Okay? So that's the second big thing that Prozac does. Exposing a zebrafish embryo can affect the stress physiology of the adult zebrafish as well. So this is another aspect of physiology that is disrupted severely by a single exposure to fluoxetine. So here's cortisol. Does everybody know what cortisol is? This is the stress hormone. Fish and humans have the same stress hormone, cortisol. Some of you may know cortisone cream. That's a kind of synthetic version of that. But basically, this is the one that's going to stimulate the liver to have more energy, help you adapt to a stressful situation, etc. So Marilyn Vera Chang is a PhD student in the last kind of year of this study, and she's lent me some of this data. And what she's done is take embryos and expose them for 6 days to the Prozac. Then she puts them in fresh water, grows them up, so 5-6 months later, and she looks at their cortisol levels and their response to stress. So if we go here, this would be the whole body, so she grinds up the whole body. So that would be the adrenal gland plus what's circulating in the blood. And she's done a study where she's exposed them to 2 levels of fluoxetine, the same as before, and she has normals or those that are stressed. And the stress is to grab the fish and keep it in a net for a specific amount of time. A standard handling stress is what it's called. And normally what you'd expect is cortisol levels to go up. And in fact, that's what happens in the females, and that's what happens in the males. And basal levels are decreased by Prozac exposure. And in females, the stress response is not altered. and we repeated this many times. In the male, you have quite a different situation. So you have a male-specific effect here. Basal levels go way down, and the stress response is reduced by about half. So they're not able to do normal stress things. Now, less cortisol is not necessarily a good thing. High cortisol can give you immunosuppression and diseases, so can inadequate cortisol levels. Because the stress, especially a short-term stress, the cortisol is an adaptive response. It helps you mobilize energy to deal with that stress. It's chronic stress that's a big deal. Chronically high or chronically low. We know that this is chronically low because it actually persists over generations. So stress physiology is upset. So here we go, reproduction, feeding, and stress physiology. Not a good situation. It also affects the behavior, and this is unpublished data. And this is called the exploratory tank behavior, and perhaps you guys are doing some of this as well. Here's a fish that's thrown into a new tank, and another fish that is thrown into a tank. Which is control and which is fluoxetine? Who says A is fluoxetine? Who says B is fluoxetine? 40-60. The answer is that this is an abnormal behavior. A fish, you throw it in, it should explore the tank. And this is quite obvious. They do explore, but they rarely go up top. This is one of the extreme examples, but they kind of spend more time on the bottom and they go up a little bit. Okay, so this is a trace of that behavior. So your upsetting behavior, we know this is linked to the decrease in cortisol, because cortisol is involved in controlling this behavior. So I call this systems control failure. Okay, a single chemical can affect reproduction, stress, feeding, metabolism, and behavior. Okay, the big question we have, and I'd love to do this experiment, is to do a population level experiment. I don't really have enough time right now, but I started to talk to people. Have you guys heard of the Canadian Experimental Lakes area? This is a huge area, size of a small European country, where they've dedicated the lakes to pollute and to study the effects of pollution. So the ethynyl estradiol study of Karen Kidd led to major changes. The guy that started it, David Schindler, was acid rain and things like that we now know have major international legislation. So I don't know if we're going to be able to do it, but I would love to be able to take a drug like Prozac and put it in the lake and see what happens over several years to see if there's population effects. Perhaps the mesocosm thing is a little bit more feasible, what you guys are trying to do with guppies. So I think it's an important thing to do, because everybody says, well, so what if it's a goldfish or a zebrafish? But that's what we do. We study these animals to figure out what's going on. But does it have an effect on the population? So I think that's a major important question in the next years. Now, here's an experiment that we've done, especially Canada and Finland and Brazil and New Zealand and Australia and all the other places that make paper. Canada has made a lot of paper for a long time, and we've been doing that population level experiment. We've been throwing phytochemicals and other pollutants into rivers and streams all over the second largest nation on Earth, and it has affected the reproduction of fish. So there's a huge survey over the last 20 years, different cycles of reviewing the effects of pulp and paper mills on fish in Canada, and downstream of fish in certain areas with certain types of pulp and paper mill factories, I guess you call them, fish have lower gonadal size. They're basically 1/4 the size of a normal fish. So these effluents seem to be affecting reproduction. And we asked the question, what's in the effluent, what's in the stock, the trees, the thing that goes into the factory, that could be explaining upsets in reproduction? So this study was really by a former PhD student, Andrew Way, and it really got us thinking about how chemicals can affect the brain, this idea of neuroendocrine disruption. And the 1 second version is that we know there's chemicals that are phytochemicals that act on vertebrate nervous tissue. So there's neuroactive chemicals in trees. We know there's neuroactive chemicals in other plants. If you smoke a joint, it's for the neuroactive effect. If you take some sort of phytomedicines, you know, they reduce anxiety and things like that. So the chemicals in plants are neuroactive. So trees as well. So I contacted some guys in industry in an arm's length organization called Forestry Products Innovations, and they're a research organization. And they do tests with fathead minnows to help the mills clean up their effluents. And here's the thing. They put males and females together, and they let the females lay eggs, and they lay eggs on a surface so you can count them easily. And then if you just let them go along, they just accumulate eggs. And they take different mills, and they take the 100% effluent. It's non-toxic from the traditional point of view. But if you look at mill C as an example, they stop laying eggs 1 day later. And this one takes a few days to shut off ovulation. They stop laying eggs, just like that. So when I saw that, I said, this is a brain event. Ovulation is a neural event. It's controlled by the nervous system. So we started working on it together, and quite a few years later, I have a story to tell you about that. And the other fun thing is that if you put these fish back into fresh water, a few days later, they start laying eggs again. So it's reversible. This is the brain. Has to be the brain. I convinced them to put money into it and all that kind of stuff. And I'll give you a little lesson on fish endocrinology and where we attack this from as another example of neuroendocrine disruption. So here is the pituitary gland. So between your ears, below your eyes, it's outside the brain. And the part of the brain, part of the pituitary that's important for this discussion is the one that produces luteinizing hormone LH. So this is the one that surges to cause ovulation. This is the one that the contraceptive pill shuts down so you don't ovulate. And in fish, it's controlled by many, many systems, same in other vertebrates. And the principal stimulator is called gonadotropin-releasing hormone. This is a small peptide. It stimulates LH. And the key one, these two are key for today, dopamine and GABA. Dopamine is a catecholamine, and it inhibits reproduction in the brain. So it controls these systems. GABA, gamma-aminobutyric acid, is a major neurotransmitter. It's synthesized from glutamate, which you probably know, by decarboxylation. And it's a major controller of reproduction as well. I told you a little bit about Prozac and serotonin, so that's why that's there. So if something in a mill effluent could alter these, it could alter reproduction. So that's the drift. That's the hypothesis. I guess the alt hypothesis is that the null hypothesis would be that there are no phytochemicals that would affect these systems. Okay, we can reject that because we know that they are there, and that's what I'm going to tell you about. So just a very brief part of the story is about in vitro assays that measure activities of enzymes that reflect the neurochemistry of the brain. So here you have dopamine, and it's a catecholamine, and it's degraded by something called monoamine oxidase. And there's a beautiful assay for this. And we found the chemicals that inhibit monoamine oxidase in those effluents. Here's a glutamate that gets converted by decarboxylation to GABA. And there's chemicals, plant chemicals, in those effluents that will inhibit this as well. So if you inhibit this enzyme, you get more dopamine. If you inhibit this enzyme, you get less GABA. So that's reproductive inhibition and reproductive inhibition. So that's the drift. So we did a survey of a bunch of different mills from 3 different countries, Canada, Brazil, and New Zealand. And these were extracts. And it's a complicated slide, but I'll take you through it. Here's the activity of that enzyme, monoamine oxidase. And these are different types of extractions. Where is that phytochemical that might inhibit this enzyme? And these two lines here, this is two different controls. This is the positive control, and this is the negative control. So what we're looking at is things that inhibit, so going towards the blue line. And you just scan that for yourself, but you can see that quite a few of the mills and quite a few of the fractions have an inhibitory function. Look at this one for Brazil. This really almost knocks it down to the clinical drug that's used to manipulate this enzyme. So this is a potent inhibitor of that enzyme system. This other ones are pretty strong as well. So no country escapes this. We don't know what the chemical is really, the chemicals are, but you find it in different trees and in different countries, okay? So there's definitely things in there that affect the dopamine system. We have lots of other data to back that up. Same with the GABA system. We're going to measure this glutamic acid decarboxylase. And again, here's the percent activity. This is the negative control. This is the positive control. So we're looking for inhibition towards the blue line. Canada, Brazil, New Zealand are all guilty. There's something in there that can inhibit this enzyme. And so there's neuroactive chemicals that affect enzymes in the vertebrate brain. Now, here's the fish, the female's there with her eggs, and we wanted to see if this is actually linked to inhibition of spawning, okay? Because that's what we would predict. So Andrew did this really interesting study, and what we found is that it actually affects the female, not the male. So here's a study he did with the FP Innovations, the animals in control, animals exposed for a week or so, and then you try to get the eggs out. How many do they lay? How many can you get out of them by squeezing them? Did they ovulate? Can you still count the eggs? And the answer is they are inhibited. So you're inhibiting ovulation. We know it's ovulation because if you look at milk production, so the sperm production in the male, it's not affected. So here's a male effluent, and basically they have milk, and they lose milk when they spawn, and they still have lots of milk. So they're spawning. You can watch them. They spawn. The males release their milk, but the females aren't releasing. The females aren't releasing eggs. So we're inhibiting ovulation. We have some other data looking at brain gene expression with that microarray I told you about, and it fits in to a major effect on the dopamine system. I think the stronger data is saying that we're intensifying that dopaminergic inhibition. And that's the conclusion from that, okay? So that's example 2. So a pure pharmaceutical agent, a pure natural compound, but we've taken forests and grind them up and thrown them out. I think we're throwing away the valuable material, all of these beautifully medicinally active chemicals. The third example is not my own work. You've all heard of bisphenol A. And this one is just a crazy one in my mind. It was designed as a pharmaceutical, but we use it in plastics. So when they discovered what was originally called estrin, which was a mixture of estrogens, then they finally isolated the estradiol and estrone and all of the other estrogenic chemicals. The chemists started making analogs to start making synthetic estrogens for contraception. And this one, I forget the date it came out, but it's like 20s or 30s or something. It's 30-something. It was invented. And it wasn't a very good estrogen. So it sat on the shelf. It didn't go into the pharmaceutical contraceptive thing. But somebody realized that it can change the quality of plastics. So this is why we're in deep, deep trouble. Bisphenol A, a weak estrogen we're putting in plastics, and we're in the 21st century where we're obsessed with plastic use. And a lot of things have bisphenol A and the bisphenol S, the new one. Let's change the bisphenol S. We haven't even tested it. Turns out to be estrogenic as well. So this one is really, really interesting because we knew that it was a pharmaceutical, but we stick it in plastics. And this is from a lab in the U.S. Jennifer Wolstenholme is the first author, and Dr. Rissman is the senior author on this paper. And this, for me, is bothersome. It says transgenerational effects of bisphenol A, abbreviated BPA. So it's a complicated experiment, but what they did here is they fed the mother bisphenol A during a certain time in pregnancy, and that was to try to get levels in the blood that a North American, a person from the U.S. would have, because bisphenol A is in all of our blood if you've been sitting on a plastic seat or around plastic or having plastic bottles and things like that. And that's not a very high level, but it's most definitely detectable. So they mimicked that, and they exposed a certain time, and then they started looking at the offspring and generations forward. So only the mom was exposed. So her offspring are what's represented here. And they measured genes in the brain called oxytocin, I told you about that one before, and vasopressin. So these are really important things for sexual behavior and other functions as well. Vasopressin, you may know as antidiuretic hormone. So it has other functions. But look at what's happening. And the color coding is here. Controls are in blue, and bisphenol are in yellowish orange, male versus female. So if you look at the part of the brain that has these neurons that express these genes, the offspring have reduced levels. In this case, it's significant. In this case, it's not. If you put them in a clean environment for a couple more generations, 3 generations, you see the same pattern. It's not as the same degree, but basically the reduced expression persists over generations. And here you see it especially in the males. So this is a genetic correlate of a lot of important functions: sexual function, osmoregulation, social behavior, and things like that. So this is affecting things across generations. Okay, here's another type of study that's very similar. This is from Cheryl Rosenfeld's lab, and she works on social mice, different species, and she has a juvenile social recognition test. It's a little complicated. I'll take you through that as well. So basically, you have a juvenile mice that are going to be introduced to an adult female. And at the very end, you replace that female with a new female. So that's basically looking at the social interaction, and as they get to know each other, they spend less time together. So they're familiar. They're not as curious. But when you put a new female in, they become curious again. So what we're going to measure is this dishabituation when you put the novel one in. So if it's a juvenile male and you put a new female in, what do you expect him to be? Extremely interested. But except if they're exposed to bisphenol A. So the way that this works is you do it on 8 days in a row, and then you put the novel female on the 9th day. Okay. So here's how it works. And the same color coding here. So males in blue, females in this orangey thing. Control versus bisphenol A. And here's what happens. The time sniffing the mouse, I guess, and then you can see the male here and the female controls. They just kind of get used to each other. It takes a lot longer for the ones that are exposed in utero to lose this interest. And this is normal. The other thing that's normal is when you put the new female in, the interest goes back up to control levels. Okay, so there's a little, you can't really see it on these figures. They're a little bit bad, but basically the ones that are the male that are exposed to bisphenol A is slightly reduced here. If you look at a couple of generations later, you have the same pattern, but in fact, this one here is the most important one. The males do not respond to the novel female. Male is not interested in a new female. And this is generations later, okay? Now, this relates to the previous work because it's decreased AVP and oxytocin. I can explain this. If you inject these things, you increase this behavior, okay? They've done gene knockouts. They know that these peptides are in controlling this type of behavior. So that's the link between the two studies, okay? So here's another really study that really hit the news there some years ago from Rosenfeld's lab again. Bisphenol A exposure makes male mice less masculine and less attractive to females. So this is the other way now. Okay, that was the male interest in female. This is the female's interest in the male. So Cheryl Rosenfeld does this kind of test where she puts a female and 2 different males, okay? And basically, they wanted to determine whether a female can sense a compromised state of developmentally exposed males. So they were exposed in the mom, and then look after birth, at breeding age, okay? Here's the results. This came out a few years ago. It's a very profound study, I think. And this particular species is doing nose-to-nose contact, and you have controls, females, and bisphenol A females here. Okay, and you have the control male and the bisphenol A male. So a control female most definitely prefers a control male. She can sense that this male is compromised. They were exposed in utero months before. If the bisphenol A female was exposed in utero to bisphenol A, she also prefers normal males. Okay? She can also note that the male was exposed to bisphenol A. So they most definitely prefer normal males. So just think of the profound nature of this result. This is, for me, this is very upsetting. That exposure in utero to a chemical, and it's probably other ones, can affect the male's ability to attract females. This is not good news for our species, nor for mice, and maybe not for others. This is how I interpret this. I interpret this in the negative. It doesn't look good. This is how Cheryl and lots of other people are interpreting this as transgenerational modification of sexual and social behavior for the negative. Okay? So the point of the lecture is really the concept of neuroendocrine disruption is expanding endocrine disruption. It's not just about vitellogenin in the liver or whether you have less estradiol or testosterone. It's really upsetting so many things. And we talked a bit about some of the mechanisms within the brain, and some of the things that are upset. We talked about sexual behavior and hormones and things, but people have shown that memory, learning, locomotion are all upset by certain chemicals, and there's so many of them here that could do different things. This can also have an impact on growth, homeostatic mechanisms, and reproduction. And I also gave you a flavor of Rosenfeld's and Wolstenholme's study with bisphenol A showing transgenerational effects. So there's a lot of people involved in this, and thank you for your attention. There's some of them that were involved in the studies that I presented. Thanks very much.